Today we're going to learn about the process of meiosis. So we're going to learn how the chromosomes get separated and put into sex cells of parents so that they can create haploid cells in the sperm and haploid egg cells as well. So to begin with, let's open up to Science Notebook page 61. And we're going to begin by titling our page here at the top with just writing meiosis. The very first stage of meiosis is just like mitosis. Mitosis and meiosis are both the process of cells dividing to make new or more cells, but mitosis happens to all of your body cells except for your sex cells. Meiosis is the process of cells dividing to produce sex cells. So meiosis, we'll write up here, equals the production sperm and egg. All right, so the first step, just like in mitosis, is interphase. We're going to highlight three parts of interphase that are important. Chromosomes are duplicated. Sister chromatids are attached to each other at the centromere. I'm going to underline some vocabulary terms that are new to us but are in our meiosis vocabulary foldable. So our sister chromatids are identical copies of a chromosome that are attached by a centromere, and that centromere is the center region that joins two sister chromatids. The third aspect of interphase that we're going to highlight is that centrosomes are duplicated. Centrosomes was another one of our vocabulary terms. A centrosome is located in a cell's cytoplasm. It organizes the microtubules used to separate chromosomes during cell division. In this case, cell division is meiosis. Now that we have interphase, the aspects of it written down, the most important parts of it, we're going to draw a picture of interphase. And on this page, we're only going to have two steps, so we can use some space here to draw our cell. We're going to draw our centrosome with the first aspects of the microtubules starting to come out of the centrosome. We have our nuclear envelope, which holds our DNA. And at this time, the DNA is still formatted in chromatin. So I'm going to draw some long, stringy looking organelle pieces here to indicate the chromatin. And we can even label this as our nuclear envelope. And chromatin. The next step is prophase. But in meiosis, prophase, prometaphase, metaphase, anaphase, and telophase all happen 2 times. So we're going to identify this as being prophase 1. In prophase 1, those chromosomes condense. The nuclear envelope dissolves. Homologous chromosomes bind to form what's called a tetrad. Homologous chromosomes is one of our vocabulary terms, which means chromosome pairs that are the same length and contain the genes for the same traits. And a tetrad is also one of our vocabulary terms, and that is a paired set of homologous chromosomes, each made of 2 sister chromatids. So let's draw what that tetrad could look like. So I'm going to be drawing the cell to show what's happening inside of the cell. But in the space off to the side, I'd like to draw a tetrad. So to begin with, I'll draw my 2 sister chromatids that are attached by the centromere. And this is one chromosome in the cell that is duplicated. So this one that's attached, these are the exact same, they're exact copies of each other. The tetrad is both of these sets of chromosomes. To show that these are two different chromosomes, I'm going to use a marker to color them in. I have an orange chromosome, and I have a yellow chromosome. Now in Prophase One, like we said, the chromosomes condense, the nuclear envelope dissolves, and homologous chromosomes bind to form a tetrad. So to show this, I'm going to draw the fact that since the nuclear envelope has dissolved, there's no longer a nuclear envelope. The chromosomes have now condensed into the chromosome shape. So here's one of my sets of chromosomes. This will be my yellow and my orange, which have formed that tetrad, meaning that they found each other. And I'll have another set of chromosomes, the blue and the green, that are also in a tetrad, and they have found one another. Still have the centrosomes, which have duplicated and are starting to go to the two poles of the cell. So to show that, I'll show the microtubules coming out from them. I'm here coloring the chromosome sets. So this tetrad is yellow and orange. This tetrad is green and blue, so we can watch what happens as this cell divides. We can chart and keep track of where the chromosomes end up at the very end. All right, time to go on to the next phase of meiosis, which will be Prometaphase 1 and Metaphase 1. So we're going to turn to page 62 next. Prometaphase will also happen twice, so we need to use that indicator that this is the first time prometaphase occurs. And in Prometaphase 1, are spindle fibers attached to the centromeres of the chromosomes. The spindle fibers, also known as microtubules. We will draw Prometaphase 1 as it already has happened once we draw Metaphase. Metaphase will happen twice as well, so we need to indicate that with the number 1. Now let's draw the spindle fibers that are attached to the chromosomes as those chromosomes are in the middle of the cell. At this point, the centrosomes have migrated to the two poles of the cell. Those spindle fibers or microtubules are attached to the two sets of chromosomes. Chromosomes are in the middle. The microtubules coming from the centrosomes are attached to the centromere of each chromosome. On to page 63. We'll start with the next phase, which is anaphase. Anaphase happens twice, so we indicate this one with the number 1. In Anaphase 1, the spindle fibers will pull the homologous chromosomes apart. And the sister chromatids will remain attached to the centromeres. Again, we're going to draw the cell with our centrosomes. But now the spindle fibers have pulled the chromosomes apart. The tetrad is no longer formed. It's on the left side of the middle of the cell. We should have one chromosome migrating to the left side, the two sister chromatids. The two sister chromatids of the other chromosome have also moved over there, whereas the other set of the tetrad is moving to the other side of the cell. These are being pulled by the microtubules, so we need to make sure to add those coming from the centrosomes. Keep track of our chromosomes. Our left one is yellow and green. Our right chromosome, they're blue and orange. After Anaphase 1, we now have Telophase 1. Telophase 1 is often, and Telophase 2 are often connected with cytokinesis, so we'll connect these two steps and record the information for both of them together. In Telophase 1 and cytokinesis, the sister chromatids will be found at the poles of the cell, and they will then decondense back into chromatin. the nuclear envelope reforms as the cell divides into two. And this is now going to result in two haploid cells. Haploid is abbreviated with the letter N, so I'll put that in parentheses. This is a vocabulary term, so I'll underline it as well. And haploid just means, as we look at our foldable, that it's a cell that contains only one set of a chromosome. So before, the original cell had a yellow and an orange, a green and a blue. But now as this cell divides into two, these two new cells are only going to have one set of the chromosome. It will either have the yellow chromosome or the orange chromosome, the green chromosome or the blue chromosome. So we will draw that down here with the two cells that are then reformed to show how these chromosomes end up in each cell. So since these two cells came as a result of splitting these two chromosomes will be yellow and green. These two chromosomes will be orange and blue. Now, this is not what it would end up looking like because the nuclear envelope would reform as these two cells separate. These also would not be in the form of chromosomes. They would be in the form of chromatin. But for the sake of showing you how the chromosomes separate into the two cells, I'm going to leave them here in the format of chromosomes. But just understand that the chromosomes will decondense into chromatin, and they will have a nuclear envelope that reforms as the cell divides into these two to help keep everything together. Let's move on to page 63. So let's show what it would look like after the nuclear membrane reforms and these two cells have separated and the chromosomes decondense back into chromatin. So in order to get these lines, I'll draw a yellow line of chromatin, a green line of chromatin, a blue line of chromatin in the other, as well as an orange line of chromatin. Now it's time to move on to the next step, which will be prophase for the second time, so we'll call it Prophase 2. Let's turn to page 64. In Prophase 2, the sister chromatids will now condense. Sister chromatids condense from the chromatin state that they were in. Also, the nuclear envelope dissolves, which I might as well show that the nuclear envelope was actually present at the end of the last step. So if we go back one page, let's just draw the fact that this could be the nuclear envelope and it has the cell that is surrounding it. And the same thing for this cell. All right, back to Prophase 2. After the nuclear envelope dissolves and the sister chromatids condense to form the chromatin, we're going to move on to Prometaphase number 2. Now, I have a feeling that you understand what's happening here in Prophase, Prometaphase, and Metaphase, and the fact that the nuclear envelope is dissolving, the chromosomes are condensing, and so we'll just show one picture at the end of Metaphase. So from Prophase 2, we move on to Prometaphase 2. In Prometaphase 2, the spindle fibers will attach to the centromeres once again. And in Metaphase 2, the sister chromatids will align in the middle of the cell. All right, time to draw what happens as a result of Prophase 2, Prometaphase 2, and Metaphase 2. So let's draw our two sets of cells. Remember, we have two cells that we need to account for. So I'll draw the two circles. There's no nuclear envelope, so I can just draw my chromosomes because they have now condensed as well. So one cell is going to have a yellow and a green chromosome in the middle of the cell. And the other one's going to have an orange and a blue chromosome in the middle of the cell. We need to draw our centrosomes. The centrosomes have those microtubules which are attached, as we wrote, to the centromeres. Now, in this step, now you can see that the spindle fibers are not connecting to two separate chromosomes that are in a tetrad. Instead, they're just connecting to one chromosome. So as these pull apart, you can envision that the two sister chromatids will get pulled apart from one another. This will all occur in Anaphase 2. So let's turn the page to page 65. Let's draw Anaphase 2. Now remember, we have two cells that we need to draw for, so we need to make sure that we're drawing our two cells. In one of the cells, we'll draw the centrosomes and the chromosomes. Draw the centrosomes in each cell. Then draw that one sister chromatid is being pulled to the left side and the other is being pulled to the right side. The bottom chromosome also got split, and one sister chromatid is getting pulled to the left and also one to the right, and the same thing happened in the other cell. These are pulled apart by the microtubules, or the spindle fibers, that are attached to the part that was the centromere, so we need to make sure that we add those in there. Now, to keep track of which chromosome is which, or which sister chromatid is which, we'll go ahead and color these in. So in this cell we have a blue and an orange. In the cell on the left we have yellow and green. Now you can envision that each of these cells will be split into two. So we're going to end up with, if we split each of these cells, we'll end up with 4 total cells, because we have 2 now. If we split both of them in half to make 2 more, then this one will turn into 2, this one will turn into 2, for a total of 4 cells. This will occur in Telophase 2 and cytokinesis. The important things that we need to note that occur in Telophase 2 and cytokinesis is first that the nuclear envelope will reform around the 4 different nuclei. And those 2 haploid cells get split into 4 haploid cells. So let's draw this occurring. Since we know that we're going to have 4 haploid cells, let's draw the fact that we have the 2 cells that kind of get cleaved and split apart from each other. To keep track of the chromosomes, we need to make sure that we're coloring them. So these 2 are going to have 1 yellow sister chromatid on the left, 1 yellow sister chromatid on the right, 1 green on the left, and 1 green on the right. The other cell will have 1 orange sister chromatid on the left, 1 on the right, and these were pulled apart from each other during Anaphase 2, so they're not with their partner. The blue sister chromatid's on the left, the blue sister chromatid is on the right. These will form 2 separate cells as that cleave continues to cleave, and that nuclear membrane will reform. So let's draw it starting to reform. And once that nuclear envelope reforms, then the DNA can condense from being in the chromosome form into the chromatin form. So let's show what it's going to look like once it condenses, the cells split, and we have 4 haploid cells. So these 2 cells and these 2 cells make a total of 4 cells. Each of these 4 cells has a nuclear envelope. And inside of each of those nuclear envelopes, we have chromatin from the chromosomes in the corresponding cell above. So this one should have yellow chromatin, and this one should have some yellow chromatin. It should also have green chromatin in each of these as well. The cells on the right should have orange and blue chromatin. And now we have 4 haploid cells. So to show that each of these are a haploid cell, let's write the letter N next to each of them to show that we have 4 haploid cells. When we started with a cell that was a diploid cell. It was a diploid cell because it had each yellow and an orange makes up the diploid cell. It has 2 sets of chromosomes, one that originally came from this person's mother, one that came from the father. Same thing down here. One of these came from the mother, one came from the father. So this here would be considered a diploid cell before it goes through its first division. So let's label this back on page 61 that this is a diploid or a 2N cell. This diploid cell ends up going through the process of meiosis and forms these 4 haploid cells. So let's draw one last diagram in order to record what has occurred over the whole process of meiosis now that we know the independent steps and what occurs in each of those steps.